Hi, everyone. I think we should get started with our next speaker. Please join me in welcoming Dr. Steve Horvath, Professor of Human Genetics and Biostatistics at UCLA. Dr. Horvath's research lies at the intersection of epidemiology, chronic diseases, epigenetics, genetics, and systems biology. He works in a variety of aspects of biomarker development with a particular focus on genomic biomarkers of aging. He's well known for his development of a DNA methylation-based age estimation method, which I think we'll hear about today, known as the epigenetic clock, or often just called the Horvath clock. Since this clock allows one to contrast the ages of different tissues from the same individuals, it can be used to identify tissues that show evidence of increasing or decreased age. So we'll hear a bit about that today, and I'm excited to welcome Dr. Horvath. It's our pleasure to have you here today. Just one comment. We are filming this session, so if you have questions, please speak into the microphone. Carrie is in the back, and she can bring you the microphone. If you raise your hand, she'll come over to you. Thank you, and thank you.
Thank you so much. Yeah. Thank you for inviting me. In this talk, I will assume that you know nothing about methylation and epigenetic clocks, but this talk is also ambitious. I would like to bring you to the very cutting edge of research in these epigenetic clocks. So I will talk about epigenetic clocks with the emphasis on plural. There are several epigenetic clocks. They have different properties. And some of them relate very strongly to lifespan prediction. Others relate more strongly to chronologic age. But before I go there, I want to remind everybody of the medieval view of mortality and death, which is this grim reaper with the hourglass. And interestingly, the modern view of mortality and aging is really another clock which is based on DNA. We should think about what precisely in the DNA molecule. In my lab, we study epigenetic modifications of the DNA molecule. So these are chemical modifications of the DNA sequence, cytosine methylation. But what I do want you to know is that the DNA molecule is really one gigantic aging clock. It allows you to track aging from prenatal samples all the way to centenarians. I want to start out with this bodybuilder who is 71 years old. And he could fool a lot of people about his age, but he, of course, wouldn't be able to fool molecular biomarkers, such as the epigenetic clock. Conversely, I show you a developmental disorder, which is known as syndrome X, because here the children never develop. They always look like toddlers. This particular person at age 20 would still look the same age like at age 2. And these children have been quite interesting to aging researchers because they seem to evade aging, or most precisely development. However, we carried out an epigenetic clock analysis of these children, and sure enough, they age as expected according to the epigenetic clock. So I want to come to a conceptual framework. In my lab, we study molecular biomarkers of aging, which are represented in the middle. And yes, they relate to these innate aging processes. I'm very interested in studying aging in people who follow a perfect lifestyle. You take somebody who lives on an island and always eats fish and has zero stress and is lean and exercises. This person will still age. The question is why? And so I call these innate aging processes. Conversely, many of you know, of course, there are many toxic exposures that people, for example, smoking, metabolic stress due to obesity, various environmental stress factors. And yes, they also affect molecular biomarkers, also, for example, epigenetic biomarkers. And so it's really difficult to build molecular biomarkers that allow you to distinguish innate aging processes from these environmental stress factors. But there's one thing that helps you, which is whatever innate aging process you study, it has to have a very strong correlation with chronological age, okay? And so when I started out with this research, I wanted to build biomarkers of chronological age. Why? Because I always hoped that something that estimates chronologic age very well probably also relates to innate aging processes, you know. So I used chronologic age in certain ways as a surrogate measure of innate aging. That's the idea. I talked about clocks, and by now many people talk about aging clocks. There's dozens and if not hundreds of articles out there on various aging clocks. But I have a very stringent definition of what I call an aging clock. It's really a marker that allows you to estimate age with very high accuracy. That a clock that isn't accurate is not a clock, right? And so I define accuracy by a very high correlation, bigger than 0.8, between the estimated value and the true chronologic age in individuals aged between 0 and 100. You will hear me talk about chronologic age. That's simply your calendar age. Whatever is in your passport, that's your chronological age. And now, if you want to build aging clocks and you know a little bit about genomics, you will come up with various ideas. There could be histone modifications, gene expression, metabolites, proteins, various clinical biomarkers, gene expression, and of course, DNA methylation, which is the topic here. Of all these biomarkers that I mentioned, of all these omics data that I mentioned, the one that allows you to do it within an hour is methylation. If I gave you a methylation data set and you know a little bit of statistics, you could develop an epigenetic clock within an hour or so. Why? The signal is so strong, you know. So let's come to methylation. To be very precise, I talk about cytosine methylation. As you know, this is one of the 4 nucleotides in the DNA. And cytosine methylation is often interpreted as an umlaut of the letter. So some people call it the 5th base. So you replace C by an umlaut. In general, epigenetic modifications are often explained as punctuation, you know, of sentences. And now, the cytosine methylation is known to play a crucial role in vertebrates. And now I highlight vertebrates because many aging researchers study other invertebrates. So they might study worms or flies. And in these organisms, there is no cytosine methylation, okay? So, however, all vertebrates have cytosine methylation, and it plays an essential role during development. An oocyte cannot develop into an embryo without methylation. Now, that's all well known, but of course, recent studies have shown that methylation also plays a role in aging, and that's very much what this talk is about. Many of my results pertain to a particular platform, the so-called Illumina methylation platform. And if you do epidemiological studies, you will know there are various versions. The oldest is the so-called 27K platform. Then there is a more recent platform called the 450K. And now the latest platform is called the EPIC methylation chip. To give you a sense, each measurement would cost you between $200 and $300. In other words, if you send me a blood sample or a saliva sample or a urine sample, you know, and you wanted me to estimate your age, it would cost maybe around $250. For that money, though, you don't just get an age estimate, you get 850,000 methylation measurements, right, on the DNA. Each measurement, each individual marker is known as a CPG, cytosine-guanine pair, And this takes a value between 0 and 1. 0 means, so let's be very concrete. You give me a blood sample, and at one location in the genome, I get a number of 0. What does that mean? It means that all blood cells are not methylated at this location. If I get a number of 1, it says 100% of all the blood cells that I profiled were methylated at this location. 0.5 means 50% of all blood cells are methylated, And so you get hundreds of thousands of these measurements at each genomic locations. Each one is a proportion, a number between 0 and 1. And the name of the game is use these variables to estimate age. In essence, it's a regression task. You use a linear regression model. So when I developed the so-called human pan-tissue epigenetic clock, I simply downloaded publicly available data. They were all available on Gene Expression Omnibus, and they were generated on these 2 different platforms. And in total, I analyzed almost 8,000 non-cancer samples. And they came from 82 individual data sets that authors had deposited into this NIH data repository. They involved 51 different tissues and different cell types. And the statistical approach was quite straightforward. It was a regression analysis where the dependent variable was age, chronologic age. And I used something known as a penalized regression model, or more precisely an elastic net regression, which automatically does variable selection. So it automatically selected 353 CPGs out of 21,000 eligible CPGs. Now, these 353 CPGs allow you to very accurately predict or estimate age based on this model. But I do want to mention that I could have removed these 353 CPGs and refit the model, and I would have ended up with another set of a few hundred CPGs, So I do want to emphasize that these clocks measure global properties of the methylome, not just specific locations, All right, so here I show you the training data that I downloaded that came from many publications. And the second column gives you the tissue, many blood, brain, buccal cells, lung, prostate, saliva, and so on. And here I show you the plot. On the left-hand side is the training data. On the right-hand side, the so-called independent test data. And this is an uncalibrated version of my clock. And what do you see? The x-axis shows the chronologic age, and you see that after age 20, there's roughly a linear relationship between the methylation changes and chronologic age. But before age 20, as the person develops, there's actually a rapid change, So we talk about the ticking of the epigenetic clock. In certain ways, during development, the epigenetic clock ticks very quickly, the fast rate of change. Now, when you use my software, you don't see that because I transform the age estimate using a nonlinear function so that you get a precise estimate of chronologic age, But you see a red curve, and this red curve is used to calibrate or transform the age estimate. As indicated by its name, it's a pan-tissue clock. It applies to all cell types that contain DNA. And it is the most accurate molecular biomarker of aging. Here I just show you a random plot I created a couple of months ago, but nobody has ever invalidated the clock. And people have looked at it in hundreds of data sets. So you always get a very high correlation between the methylation age and the chronologic age, as long as your samples are have a good age range. If you have children and older people in your study, you will get a very high correlation. Of course, if you study a birth cohort where everybody is exactly 40 years old, the correlation will be, you cannot even calculate the correlation, right? what people have observed is that the pan-tissue clock underestimates ages in older individuals. So here I show you how the clock performs in centenarians and supercentenarians, and you see it underestimates the ages. And in general, people sometimes observe offsets, and offsets could very much correspond to biological differences, but they could easily also depend on technical aspects. So, for example, maybe the way the DNA was stored, or the bisulfite conversion treatment. And so we are currently finding ways of minimizing this technical variance, so stay tuned for that. A lot of my results were obtained in blood, but I do want to emphasize the clock applies to all cells. So here I show you bone cells, osteocytes, osteoblasts, and you see very high correlations. Any cell you can think of. Let's give you a typical example of a study in humans. So one of the first findings was that obesity actually accelerates epigenetic aging in the liver. And I want to show you how one can demonstrate that. The upper panel shows you how the methylation age on the y-axis relates to chronologic age in humans. And what do you see? You see high correlations in liver, adipose, muscle, and blood. That simply says, well, the clock measures chronologic age. However, you also see a dashed line. This is a regression line. And think about the meaning of that line. It's really the expected value. If a point is above the dashed regression line, it means the liver sample is older than expected. And if the dot is below the line, it means younger than expected. So this discrepancy or this difference between a methylation measurement and the regression line has a name. It's called age acceleration measure. It's a residual from a regression line. And this residual, you want, of course, a negative value because it would indicate that you are aging or your liver ages more slowly than that of your peers from the same age group. So this age acceleration measure takes values between negative values and positive values. Value 0 means average. And in the lower panel, I show you how epigenetic age acceleration on the y-axis correlates with body mass index. And what you observe is for liver, there's actually a pretty strong correlation, around 0.4. And that's the statement. People with high body mass index, their liver has a higher epigenetic age than expected. Conversely, look at the blood samples on the far right. There, the age acceleration doesn't really correlate with body mass index. And that makes already one important point. Many stress factors, such as metabolic stress, have cell type specific effect. So they might age hepatocytes in the liver, but they may have a relatively weak effect on blood cells. And to tell you, we, of course, carefully evaluated blood in much larger studies, in thousands and tens of thousands of people, and then we do observe a correlation between body mass index and epigenetic age acceleration. However, it's a much weaker effect. Correlation maybe 0.1, Here's another finding. People often ask, do organs age at different rates? And one example I want to cite is actually female breast tissue. Turns out that female breast tissue, for reasons that are not well understood, seems to be older than other parts of the body. And I looked at 30 different body parts in people, and female breast tissue really stands out. Here I show you data from a study where we had 80 breast tissue samples and 80 matching blood tissue samples from 40 healthy female participants, meaning none of these women had cancer, The red dots show you the ages for the breast tissue samples. The black dots show you the results for blood. What do you see? The red dots always lie above the black dots. So, and even in young women, do you see these? Some of these women were in the early 20s. So it's an interesting insight, which perhaps points to a role of hormones when it comes to epigenetic aging. I want to talk about new human clocks. And one new clock was developed really for in vitro studies. And we call it now the skin and blood clock. And here's the original motivation. So the original pan-tissue clock led to large errors in fibroblasts. Mind you, there's a high correlation, right? A correlation 0.92. So if this was a regular biomarker of aging, people would be happy about it. However, when you look at the error, the median error was 12 years. So I was always unhappy about that result. Why? Because people use fibroblasts and cell culture systems for preclinical work and really deep biological studies. So I built a new clock, the so-called skin and blood clock, which is really tailor-made for human fibroblasts, keratinocytes, buccal swabs. You give me a buccal swab, I can estimate your age very accurately. Endothelial cells, blood, and so on. So it's so accurate that you could use it for forensic applications. Having said this, it has a limitation. It's actually not the best predictor of lifespan based on blood. So yes, it predicts how long you live, but it's not great at that. We have much better clocks for that. And let's step back. I think nobody disputes that the epigenetic clock is the most accurate molecular marker of chronologic age. However, there is a debate, well, to what extent does it relate to biological age? And many people will talk about biological age, but to tell you, pretty much everybody has their own definition. You know, people talk about it as if it was a well-defined term, but it isn't. Now, what everybody agrees on, though, is that whatever definition of biological age you have, it should have a couple of properties that are listed here. So first of all, your measure of biologic age better predict mortality or lifespan even after you adjust for chronologic age and a list of other covariates, such as sex and maybe various confounders, such as a prior history of disease. The other property is that if you analyze the offspring of centenarians, then this biomarker of biologic age should actually be younger in those than in others. In other words, if your parent lived to age 110, chances are you inherited good genes or whatever. Therefore, probably your biologic age is younger than that of your age-matched spouse, you know, whose parent didn't live that long, you know. And the other criterion is that whatever measure of biological age you have, it should really relate to many age-related diseases. Not all of them, but many. I mean, chronic diseases and various measures of frailty and so on. And I can tell you that the epigenetic clocks, they satisfy these criteria, and I will flesh that out. Let's discuss it one by one. Does DNA methylation age predict lifespan due to all-cause mortality? And the answer is a clear yes. There's dozens of independent studies by independent groups have shown that. I mentioned a large-scale meta-analyses in the US, a Danish twin study, a German study, and so on. So everybody has validated it. In my lab, we did the largest meta-analysis study, and the large list of authors indicates that we used many cohorts, 13 cohorts, over 13,000 individuals. And I want to describe the data to you. So these cohorts, such as Women's Health Initiative, a Framingham Heart Study, Lothian birth cohorts, and so on, these cohorts collected blood samples back in the 1990s or earlier. And they put these blood samples in a freezer. And so but next they followed the people. So they knew whether the person was still alive 20, 30 years later, or they knew more. They knew what age they deceased, what was the cause of death, and so on. And so you can then generate methylation data from the blood samples from the 1990s and see whether that predicted how long people ended up living and what they died from, and so on. And that's what we did. So here I show you a typical plot that you get when you do these meta-analysis studies with a Cox regression model. Focus on the p-values. So here I give you, in certain ways, the worst p-values. Why? Because this is lifespan prediction after you adjust for every possible confounder we could think of. We adjusted for chronologic age at time of blood draw, body mass index, education, alcohol, smoking, history of diabetes, cancer, hypertension, physical activity. Anything that we knew relates to lifespan, we put in the regression model. And even after you adjust for all of it, it still predicts lifespan. That's a very powerful hint that these epigenetic clocks do not just measure chronologic age. They measure more because they can predict something, even after adjusting for confounders. I want to move to our most recent and best lifespan predictor, which we have named after the Grim Reaper. It's called DNA methylation Grim Age. It was constructed in a roundabout way, and you might learn something from the way it was constructed. That's why I go into the details. So it was constructed in 2 stages. In stage 1, on the left, we first developed methylation-based surrogate markers for various plasma proteins and smoking pack-years. So, for example, some plasma proteins are very exciting to aging researchers or clinicians. Plasminogen activator inhibitor 1, growth differentiation factor 15 in the context of senescence, and so on. Now, interestingly, so we built methylation-based estimators of plasma proteins, which is kind of a crazy idea, because normally you would measure plasma proteins using an immunoassay or another technology. But anyways, we did that. Also, we built a methylation-based biomarker for estimating your smoking pack years. You give me a blood draw, I tell you how many cigarettes you smoked in your entire life, That's your smoking pack year history. And now we have these methylation-based biomarkers, and next we, Arkelu, fit a regression model, a Cox model, to predict lifespan. Do you see? That's in stage 2. So the resulting biomarker is 100% based on methylation measurements. It doesn't use protein measurements. However, it does predict time to death, and it's really a hazard ratio. And you would get an age estimate. If you give me blood, I would give you your grim age. So, for example, I celebrated my birthday half a year ago, and I turned 51. And then I measured my grim age, and I was very happy that it was only 49. Okay, so and I have an identical twin, and I measured his grim age as well. And up to the decimal point, he had the same age, 48.9. I mean, it's amazing. Twins, so identical grim age. So now, Grim Age is named after the Grim Reaper because it's very strongly related to lifespan. Here I show you the meta-analysis p-value 10 to the minus 75. That is such a significant p-value that yes, there's a chance that it's a wrong finding, but that chance is lower than the probability that an asteroid will hit planet Earth tomorrow and wipe out all life forms on this planet, you know. So it's a very significant p-value. But also, and that Grimm age predictor applies to all groups you could think of. Apply it to women who never smoke. Apply it to men who are heavy smokers. Apply it to Hispanics. Apply it to African ancestry. Apply, you know, all groups, carefully validated. It also, interestingly, relates to a lesser extent to time to coronary heart disease. So you see the second p-value. But also, surprisingly, time to cancer. And that's surprising because you take a blood draw from a person who has no sign of cancer, and it still seems to predict time to onset of cancer. Lifestyle factors are associated with the lower age acceleration. Remember, by the way, age acceleration denotes measures that are adjusted for chronologic age, you know. So we measured vegetable intake using mean carotenoid levels, and you see these p-values. Do you see mean carotenoid levels have a correlation of 10 to the minus, a correlation minus 0.26 with Grimm age in blood? To me, that's a strong correlation. Markers of inflammation, C-reactive protein, or markers of various markers of metabolic syndrome, insulin, glucose levels, all relate in the expected way with Grimm age. Fatty liver disease is very much related to accelerated Grimm age in blood. At the bottom, you will see some variables that are interesting to social scientists. Education. No question, higher levels of education are associated with lower epigenetic aging, p-value 10 to the minus 9. However, the effect is weak. Do you see the correlation in this case is only minus 0.09? That's a very weak correlation. And you only observe it when you study thousands of people. Like in this case, we studied 4,000 people. Another variable that's even more is frustrating is to see there is even a relationship to income levels. The correlation is weaker, but there is one. Physical exercise protects you from epigenetic aging, correlation minus 0.1. Look at smoking status, correlation 0.4. So smoking is by construction very strongly related to Grimm age. What about alcohol consumption? There, the p-value is only 0.02 when you study 3,700 people. However, the effect goes in the direction drinking is good for you. Do you see? Correlation minus 0.04, you know. So this is meant as a joke, you know, but a correlation of 0.04 minus 0.04 means there is no effect, you know, but yes. Yeah.
So these correlations are kind of hard to interpret. Have you done some regression analyses where you try to estimate, if you will, the effect? And I put that in parentheses because it's hard to know what effect is of education, a year of education on Grimm age, and compare that to some other types of outcomes. So rather than correlations,
yes,
kind of partial effects.
Yes. So the comment is that these are correlation tests, which in certain ways hide the effect size of, let's say, one additional year of education, you know. And so definitely in our papers, we fit these multivariate models, you know, that allow you to estimate these effects. Absolutely. It's often hidden in the supplementary information. You know. What I do want to mention is, yes, healthy lifestyle is good for you. Trivial comment. But the other comment is these effects are actually all weak, you know, and except for smoking, you know. And so these, and that could reflect the tissue, because maybe if we measured markers of metabolic syndrome in liver, there would be much stronger correlations, you know. So be mindful of what tissue you study. So methylation biomarkers have been linked to many conditions, and I will get into them. Here I list them in alphabetical order, from Alzheimer's disease, cognitive decline, education, heart disease, HIV status, Huntington disease, obesity, pubertal development, progeria such as Werner syndrome, and so on. What you do want to be careful about when you study the literature is you want to be mindful, where was the association found, in what tissue? Because, for example, it's very difficult to show a relationship between Alzheimer's disease and epigenetic aging in blood. There might not be one. I don't think there is one. However, in prefrontal cortex, you do see associations, do you see? So what do epigenetic clocks teach us about anti-aging strategies? I'm, of course, an aging researcher, and that's kind of interest to me. I briefly talk about it, but then later we talk about social science applications. One anti-aging strategy is cellular reprogramming, and more precisely talking about interrupted cellular reprogramming. So let me explain what that means. You may know that Yamanaka won the Nobel Prize for showing how to use adult cells, for example, skin cells, and turn them into stem cells, so-called induced pluripotent stem cells. And that procedure, the so-called Yamanaka factors, completely reset the epigenetic age to a prenatal stage. That is a rock-solid finding that has been confirmed in dozens of applications. So you, in certain ways, if I apply these Yamanaka factors to you, I can turn you into an embryo, you know. However, that's, of course, not the idea, you know. So here, when you carefully look at the time course experiment, you will notice that this epigenetic age on the y-axis actually seems to decrease with days post Yamanaka factor induction in an almost continuous fashion, okay? And there is the idea. What you do is you apply these Yamanaka factors for a relatively short time, 5 days, 6 days, so that you rejuvenate the cells. The epigenetic age is decreased by 10 years. However, the character of the cells did not change. The cell status didn't change. Why? Because you don't want to turn people into a collection of stem cells, right? For one thing, it increases your risk of cancer and so on. What you want is you want the rejuvenation effect, the anti-aging effect, but the cells don't transform at all, you know. And here I show you results from a Stanford team, Vittorio Sebastiano, where we analyzed this idea in human fibroblasts and in human endothelial cells, and we showed if you apply this transient or interrupted reprogramming idea, then yes, you actually rejuvenate the cells. Let's talk about another anti-aging strategy. It could be called organ transplants. Let me describe it. So several people have shown that hematopoietic stem cell transplantation rejuvenates the blood to the age of the donor. So hematopoietic stem cell transplantation is used when treating a very severe form of leukemia that necessitates the removal of all blood cells and then a donor transplant. And here Shigemi Matsuyama and others showed that even after 17 years, the age of the reconstituted blood is that of the original donor. So it shows that epigenetic age is a cell intrinsic property in, at least in the, when it comes to this hematopoietic stem cells. In short, if you want, one strategy to get new blood would be you grab a very young person and harvest their stem cells and transplant it. And then the blood reconstitutes itself, and the blood will have the epigenetic age of the donor. There is, of course, a problem with that beyond all the ethical concerns, and that is that this is a very dangerous procedure. It's really a treatment of very last resort, not the least because of graft versus host disease, which is associated with increased epigenetic aging. So unfortunately, that's not a viable strategy. But here I show you the data. You see on the y-axis is the methylation age of the recipient, often many years after the transplantation, 10 years later. And on the x-axis is the age of the donor, not the age of the recipient, And you see very good correlations. Strategy number 3, anti-aging strategy, hormone treatments, hormone treatments are obviously controversial. I do want to mention, though, a finding. We analyzed buccal epithelial cells, so these are collected with a buccal swab, and we found that women, postmenopausal women, who had undergone menopausal hormone therapy, their buccal cells were substantially younger than women who hadn't done, who didn't get the hormones. But there is a wrinkle to the story. Notice how I emphasize buccal cells, because we didn't see this effect at all in blood cells. And this makes sense because buccal cells have estrogen receptors, and in certain ways they're more susceptible to hormone treatment. By contrast, blood cells have far fewer estrogen receptors. So that's a way then, if you care about young buccal cells, hormone treatment. I need to emphasize hormone treatments are controversial because they may increase your risk of various cancers and other diseases. Conversely, in the data, we had also blood samples from women who, for various reasons, needed to undergo what is known as surgical menopause or bilateral oophorectomy. Unfortunately, these women had various bleeding disorders, or for whatever reasons, this procedure had to be carried out. And when we then looked at their blood samples, we found that, yes, there was an age acceleration effect. So now the good news is that these effects are all weak. So women who enter menopause earlier than expected, yes, they age a bit faster, but it's really a very small effect, at least in blood. Yes?
Did you happen to look at women who may have gone through surgical menopause but then went on to HRT, to have HRT?
In my opinion, all of these women must have received HRT, because these women were typically in their 30s, and so having said this, I did not look at that, Obviously, one would assume that the hormone treatment benefits, and alleviates that problem. Yes. Strategy number 4. Some of you may have heard of rapamycin, or there are various compounds that are quite interesting to aging researchers. And we evaluated the effect of rapamycin in keratinocytes in the dish. And sure enough, rapamycin slowed epigenetic aging in keratinocytes. Now I mention again the cell type, skin cells, because this effect we have no evidence that this effect translates to other tissues or organs, so be cautious. So I mentioned already the most boring strategy of all, follow your healthy lifestyle, and I mentioned that, so problem is that's just not effective enough, You can eat as many vegetables as you want, you will still age and die, unfortunately, so and yes, go ahead.
I was just wondering, do we know whether associations between these epigenetic clocks and mortality or morbidities are comparable among people who have had them sort of artificially reduced or increased by those first 4 strategies? Or are we sort of just assuming that given the general association of these clocks with mortality, that if they change, we assume that mortality would change similarly?
Yes, this is a very important question. It's a critical question. The entire field wonders whether these epigenetic clocks are a good surrogate marker for anti-aging strategies. So in other words, coming back to the hematopoietic stem cell transplantation, or something drastic, if you have ideally, if you actually have a safe procedure, forget HSC transplantation. If there's a safe procedure to reset the epigenetic age, let's say of blood, would that translate into benefits in terms of health span or lifespan? And the answer is nobody has done these experiments, And now in my lab, we are doing careful studies in model organisms. I'll talk about it later, One needs in certain ways model organisms that don't live that long. Because to really answer your question, ideally one has a prospective study where you administer your treatment to people in their 50s and you follow them for 40 years and you say, whoever's age was rejuvenated, epigenetic age was rejuvenated, they ended up living, And you can imagine these are expensive and time-consuming studies. And so therefore one really needs to go to model organisms. Yeah. Other questions? Yeah, it's a big caveat, Sorry. There's another caveat about these clocks, which is it is difficult to understand the molecular causes and consequences of epigenetic clocks. The good news is the brightest researchers are working on this question. Why? So what molecular mechanisms give rise to epigenetic aging effects? Or conversely, if somebody is epigenetically older, what are the downstream consequences of that? So hopefully I will be able to tell you more next time you hear me speak. However, I do want to mention already a couple of things. First of all, these different clocks have different interpretations. The Grimm age clock, for example, very much relates to immunosenescence, inflammation, changes in blood cell composition, such as naive T cells, and very weakly to telomere length. But other clocks, for example, the pan-tissue clock, seem to capture properties of stem cells, hematopoietic stem cells. So they're more cell-intrinsic properties. So there's not one explanation that fits all at the moment. What I do want to mention is that the epigenetic clocks are not a marker of cellular senescence per se. And there are several reasons for that statement. One is, when Ken Raj immortalized fibroblasts or keratinocytes, he observed that the epigenetic clock keeps ticking. So you may have heard one can immortalize cells in the sense of you prevent replicative senescence, you prevent the Hayflick limit. However, these clocks, their epigenetic age continues to increase. Oops. Some people have thought that these epigenetic clocks measure what is known as mitotic age, the number of cell divisions, for example, of stem cells. And that may be true in certain situations, but it's not true in all situations, because the epigenetic clocks work beautifully in sorted neurons, in post-mitotic cells. So a panel in the middle shows you this very strong correlation of 0.94 in sorted cells. In general, the epigenetic clock works on all brain cells, glial cells, and so on. I want to switch gear and talk about early development, because when you hear about mortality prediction, you think of aging, right? However, I could have given the same talk and not mentioned aging once. I could have just talked about human development. And one important study along these lines is in fetal retina with Tom Rhee. The fetal retina development has very little room for noise or entropy or whatever you think of as age-related decline. It's carefully orchestrated. However, as you see from the scatter plot, you get a correlation of 0.8 when you correlate methylation age to fetal age, And so in essence, the epigenetic clocks must relate to processes during development. And there's a very nice article, synchrony and asynchrony between epigenetic clocks and developmental timing. I don't want that you walk away thinking that these clocks are simply measures of developmental timing. There's also asynchrony. But there's, the conclusion of the study is that, yes, these mechanisms that underline the phenomena of epigenetic aging might actually be studied in developing organs in the dish, in organized cell cultures, and we are doing that. Coming back to human epidemiological studies. So here are typical studies where people followed a cohort. The In-Chianti cohort followed Italians. And for most people, there were 2 or 3 blood draws available. And you can ask a very basic question. Imagine you have a 20-year-old and you measure the epigenetic age in the 20-year-old, and it says this person is actually 30 according to the epigenetic clock. Now you do the same measurement 10 years later. Question is this person still much older than expected? And the answer is yes. Okay, so this epigenetic age acceleration is very stable across the entire lifespan. So, in other words, if you have epigenetic age acceleration as a teenager, you will have epigenetic age acceleration as an 80-year-old. It's quite stable. And it's linear. For most people. And why is that? Well, because epigenetic aging is very much under genetic control, development, So if you have certain genes that predispose you to faster epigenetic aging early on, it's very hard to reverse this effect. Now, if you carefully look there on the right panel, you see some curves that go down drastically. Do you see that here? And so age reversal is really probably due to, this is simply a measurement error, so maybe the DNA quality wasn't good, it's a technical artifact. I want to say for 95% of people, epigenetic age simply tracks changes in passage of time. All of that gives rise to what I call an epigenetic clock theory of aging. So these clocks, the methylation age, is really a continuous readout that links processes that play a role in development to then processes in middle age, tissue homeostasis, tissue maintenance, prevention from malignant transformation, to then ultimately processes that are more associated with organ dysfunction and aging. Many people have proposed similar theories, but what is new here is that the methylation clock really links these processes in a continuous fashion. And so in certain ways, it links purposeful molecular processes early on during development and tissue maintenance to unintended adverse consequences later in life, which is well within the framework of antagonistic pleiotropy. So coming back to the study of diet, exercise, education, I mentioned that these measures, what's new here? In this publication, we distinguish different measures of age acceleration. You see so-called intrinsic age acceleration and extrinsic age acceleration. IEAA stands for intrinsic epigenetic age acceleration. What does that mean? These changes are not due to changes in cell composition, cell intrinsics. These are properties of stem cells. Extrinsic epigenetic age acceleration, by contrast, very much relates to changes in blood cell composition. So, for example, it does measure the changes in senescent or so-called exhausted T cells, or in changes in naive T cells, and so on. So be aware of it. And remember how I told you that education, income, and healthy lifestyle and so on is good for your epigenetic clock. What I should have mentioned is it's good for clocks that measure extrinsic epigenetic aging. In other words, clocks that do relate to changes in cell composition. By contrast, clocks that are independent of cell composition show much weaker relationships with these confounders. I want to now talk about some topics that are interesting to social scientists. What about an analysis of race, ethnicity, sex, and various risk factors? Let's start out with the expected result that women age more slowly than men, according to pretty much all epigenetic clocks. That's a very robust finding. The effect is not big in blood. So women, the methylation age in the blood tissue of a woman is maybe 1 or 2 years younger than that of an age-matched man. There might be much stronger effect in liver. We also looked at brain tissue, and even there do you see an advantage for women, but there this effect is much weaker, So, yes. What about different groups? I analyzed Hispanics versus Caucasians. And so here, the upper panel shows you the methylation age versus chronologic age in 3 different large cohorts. And blue dots are Hispanic ancestry, gray dots are Caucasian ancestry or European ancestry. And you can barely see it, but if you do an analysis, you will find that, interestingly, Hispanics have a lower epigenetic age than age-matched Caucasians. And that is particularly true for what is shown in the lower panels, the so-called intrinsic epigenetic age acceleration. So epigenetic aging that is independent of changes in blood cell composition, there Hispanics seem to have an advantage. And this finding is rock solid. Here I show you validation data. These are all large-scale studies, thousands of people. So there is no doubt, according to the intrinsic epigenetic aging clocks, Hispanics age more slowly. And when I first analyzed these data, I hated it because I wanted that the clock works on all people, right? And so there's a bias. So I hated it, and I emailed my collaborator, Michael Gervin, and he pointed me out this famous Hispanic mortality paradox. Some of you may have heard of it. Back in 1986, Marquides coined the term, which refers to a curious demographic observation, which is that Latinos in the US live longer than non-Hispanic whites. So on average, a Latino lives 3 years longer than a person of European ancestry, strictly European ancestry. And back in the 1980s, this was a very perplexing finding, and people spent decades thinking of ways how hidden biases slipped into the data. And there was a large literature that in the end established that finding without a doubt. Hispanics live longer. It's not some artifact of the data analysis. Well, I, of course, think it's interesting that intrinsic aging in Hispanics is also decreased, right? Now, this is not a rigorous analysis, but it's quite a coincidence. So if you are courageous, you would say, well, maybe Hispanics have a mortality advantage because their epigenetic clock ticks more slowly. There will be caveats I mentioned later. Let me go to another population, the so-called Tsimane Indians. This is really a study by Michael Gervin from the UC Santa Barbara. So the Tsimane Indians are a population that live in the Bolivian jungle. And here I show you some figures or photographs of their lifestyle. They eat plants, but also, of course, animals. So why are aging researchers interested in the Tsimane? First of all, their ecology is typical of pre-industrial societies. High pathogen burden. They live in the jungle, right? Energy limited, limited food, natural fertility, minimal healthcare access. However, there was a paradox, which is that although these individuals have high levels of inflammation, high levels of infections, their risk factors for cardiovascular disease were much reduced. And that runs counter to a famous principle in aging, which is the so-called inflamm aging. You may have heard one of the major theories of aging is that inflammation causes aging. Now, these individuals have very high levels of inflammation, but according to various readouts, they actually have very low cardiovascular disease. They don't have these chronic diseases. They don't have atherosclerosis or various chronic diseases that plague the developed world. So there's, again, a paradox. Why does high inflammation not lead to major cardiovascular disease? Well, we analyzed the blood samples, and sure enough, very much what we had observed in the Hispanic ancestry samples, that c-MANE had much slower epigenetic aging according to the cell intrinsic clock. And now, so that if you are courageous, you could again come up with the hypothesis that maybe the c-MANE have decreased levels of cardiovascular disease and chronic diseases because they have a genetic advantage. Their intrinsic epigenetic aging is diminished. As I mentioned earlier, we distinguish two major types of measures of age acceleration. The first is called intrinsic, which is not related to changes in blood cell composition, and extrinsic, which is related to changes in blood cell composition. The extrinsic measure very much relates to measures of inflammation and so on. And I mention it because if we now study the Hispanics in the upper panel and the c-MANE Indians, according to the extrinsic measure, which is confounded by blood cell composition, you see the opposite from what I showed you for the intrinsic measure, which is, look, Hispanics actually have higher extrinsic epigenetic age acceleration, which reflects, of course, higher levels of inflammation. And also the c-MANE have much higher levels of extrinsic aging. So that's kind of the paradox, right? Because in terms of when you study Hispanics or c-MANE in terms of markers of inflammation, they look like they're in deep trouble. But Hispanics live much longer, although they have these higher levels of inflammation. But the intrinsic measures show kind of the opposite. All right. Any questions at this point? Okay, I will now come to another chapter. I mentioned centenarians. Aging researchers are very interested to find out why do some people live 110 years old. And so this was a study in Italian semi-supercentenarians and their offspring. So Claudio Franceschi and his team looked at blood samples from offspring of people who were 105. And sure enough, they aged more slowly. So that was kind of nice. I mentioned Alzheimer's disease. We analyzed a very exciting study, the so-called religious order study, that included post-mortem brain samples from Catholic priests, nuns, and brothers across the US. And these individuals were so generous to allow cognitive assessments before death and monitoring, and then they donated their brains to research. And so we analyzed 700 prefrontal cortex samples. And what we found is that, yes, epigenetic age acceleration in the prefrontal cortex is very much associated with measures of brain pathology, amyloid load, neuritic plaques, diffuse plaques, PHF tau tangles, and so on. So that was expected. Interestingly, even if you related epigenetic age acceleration in these post-mortem brain samples to pre-mortem cognitive assessments, such as memory function, global cognitive functioning levels, there were associations. Now, the p-values are much weaker. These are very weak associations, but still stunning that one observes it. So, let's switch gear. So-called progeroid syndromes. You may know progeria are in certain ways accelerated aging diseases. And George Martin, back in the 70s, assembled a list of what he considered accelerated aging diseases, and he ordered them by clinical manifestations. It turns out a condition which Down syndrome is actually the ultimate progeria according to his ranking. Now, most people wouldn't think of Down syndrome as a progeria. However, if you study various organ systems, brain, heart, and so on, actually Down syndrome individuals, unfortunately, have much higher burdens of pathology. Other progeria that are more famous are Werner syndrome or Hutchinson-Gilford progeria. Now, I'm, of course, interested in all of them. And let me tell you, the number one condition associated with increased epigenetic aging is Down syndrome. We observe the strongest effect, both in blood and in brain tissue. So yes, Down syndrome, the number one progeria, according to George Martin, shows actually the strongest association with faster aging. What about Hutchinson's-Gilford progeria? This is the progeria you know from your TV news program. So these are unfortunately children who have that condition. And we analyzed fibroblast samples from progeria children. And sure enough, we observed age acceleration according to the skin and blood clock that I mentioned earlier. Why? That clock is tailor-made for fibroblasts and in vitro studies. However, when I applied my original pan-tissue clock to these fibroblast samples, I didn't see this effect. So I'm a bit nervous about this finding because it might very well depend on the epigenetic clock that you use. What about Werner syndrome? That's so-called adult progeria. There we do see a pretty strong age acceleration effect in blood according to various clocks. And here, even in small sample sizes, here we only had 18 Werner syndrome individuals, and we see age acceleration effects. Huntington's disease. I work very much on Huntington's disease, and interestingly, I do observe age acceleration in Huntington's disease. So in my worldview, Huntington's disease can be interpreted as another progeria. Parkinson's disease, not so much. We found evidence for increased epigenetic aging in the blood of Parkinson's disease patients, but that result is really requires validation. I'm nervous about it. However, there was another finding which has been validated over and over, which is granulocyte counts. So we looked at when you have methylation data, you can actually impute blood cells. So you give me a blood methylation data set, I can tell you how many neutrophils are in your blood or how many T cells, And so we used these imputed measures of neutrophils or granulocytes to find a very strong association between PD and a neutrophil. Here's a logistic model. So I mention it because when you generate your methylation data, you can do a lot of things. Never mind epigenetic clocks. You can relate blood cell composition to your phenotype. And that may very well be the number one finding of your study. So here, for example, granulocyte counts, when you look at p-values after you adjust for everything, still highly significant. Let's talk about children again. There's the Avon longitudinal study where the people evaluated child development, right, from cord blood sample at age 0 to samples at age 7 or later teenage years. So there are methylation data from various time points. At the same time, people measured various growth characteristics, weight gain, height gain, and so on. And you can ask, are there relationships? And yes, there are. Now, these are weak relationships, but I just want to present them. Higher age acceleration at birth, meaning if you have a cord blood samples and the age is a bit higher, that is actually associated with higher fat mass at children. So what's the message here? Age acceleration early on is actually good news, because it kind of tracks development. You want that your baby has age acceleration. It's just later in life you don't want age acceleration, And so, interestingly, in boys, higher age acceleration at birth was actually associated with higher odds of increasing Tanner stages, which is, the Tanner stage is a measure of puberty, So that's in boys. However, people also reported these associations in in girls. So epigenetic age acceleration in blood is associated with pubertal timing, for example, adolescent breast composition in girls, So I invite you to look at that. I want to briefly mention in infectious diseases, one finding that has been replicated over and over again in the literature is that HIV infection accelerates age, especially in blood, but also in brain. So here I show you results from various brain regions. But in blood, it's also associated. What about things like sleep disorders? So Jude Carroll looked at measures of insomnia and blood methylation data. And so she found that various symptoms of insomnia are associated with increased extrinsic age acceleration. Remember, extrinsic age acceleration relates to inflammation and so on. And so, yes, there was an association in the Women's Health Initiative. And that persists even after she corrected for various confounders in a multivariate model. So I hate these findings. Sleep disorders are not good for you. Interestingly, in her study, she found the strongest effect again for these imputed cell types. Remember, methylation allows you to estimate naive T cells or exhausted T cells, and that so it very much tracked that. Some of you will be interested in various measures of stress, from post-traumatic stress disorder to cumulative lifetime stress. There's really a large literature. I just want to acknowledge here one finding where another group really found that cumulative lifetime stress, meaning stress over decades, is associated with faster epigenetic aging. By contrast, this short-term stress you have, this post-traumatic stress, or you go through a divorce, or this short-term stress does not seem to have an effect on epigenetic aging. It's really decades of stress. I gave you a long laundry list of everything related to epigenetic aging, but let me mention there are market failures, so to speak. Everybody knows that radiation is bad for you. It induces senescence, right? And interestingly, at least this strong radiation, let's say 10 gray, 5 gray, we never observed that this strong radiation accelerates epigenetic aging, Having said this, these are in vitro studies because in other studies, when we looked at the effect of radiation therapy in breast cancer survivors, interestingly, there we did observe effects. So in vivo, radiation might accelerate epigenetic aging of blood. It's just not in vitro. Before I go, I want to digress and mention that currently I develop many epigenetic clocks in mammals. This is a project funded by the Paul Allen Frontiers Group. We have super accurate clocks in mice. Here's just a random finding. High-fat diet accelerates aging in mouse livers. I study very exciting animals. You may know some bats can live 40 years. And here I show you centenarians, quote unquote. So these are 40-year-old animals, bats. And it's a mystery. Why do bats live 40 years, but a mouse lives 3 years, right? Think about it. What's the molecular reason for that? And so, yes, we're studying it. So we have very accurate clocks for 17 different bat species. So next time you hear me talk about it, I'll describe the findings. I also mentioned we study pure breeds of dogs in a collaboration with Elaine Ostrander from the NCI. So these are show dogs. And some dog breeds live long lives, others short lives. So here we have, I just mentioned, we have very accurate clocks for dogs. If you give me a blood sample from your dog, I can tell you how old it is, you know. So correlation across 51 breeds, correlation 0.96, you know. What about telomere length? In the past, telomere length and even today is a very important molecular biomarker of aging. And here I show you data from the Framingham Heart study where we related mean TRF, telomere measurement, to chronologic age in the upper panel. And you see a correlation of minus 0.27. That's a famous statement that telomeres shorten with aging. But for the same individuals, we also had methylation data. And the lower panel shows you then the correlation with methylation age, correlation 0.81. So this study and really dozens of others have shown without a doubt that yes, epigenetic clocks have a much stronger relationship with chronologic age than does telomere length. And now there's some literature by independent groups, but also our groups that show that epigenetic age and telomere length tend to be independent predictors of mortality. So if you have a regression model and you have an epigenetic age estimate and telomere length, both would be significant if your sample size is large enough, independent of each other. However, I do want to mention that there is no comparison when it comes to lifespan prediction. Biomarkers such as Grimm age in a linear model will give you a p-value 10 to the minus 30, and in the same model, telomere length will have a p-value 0.01, okay? There's simply no comparison when it comes to lifespan prediction. In general, methylation age acceleration is actually not correlated with telomere length. Certainly the pan-tissue clock is not correlated at all with telomere length in large-scale epidemiologic studies. And we have looked at it in maybe 10,000 people. So one would then conclude that epigenetic aging is just a different hallmark of aging from telomere shortening. Except there was an interesting finding. When Ake Lu in my lab did a genome-wide association study of epigenetic aging rates in blood, she found that one of the top GWAS hits was actually the TERT locus. TERT is part of telomerase, you know, so which maintains telomeres. So this came out of nowhere, you know, so interesting finding. But the association was paradoxical. In the sense of if you won the genetic lottery according to the telomere clock, meaning your telomeres are longer, you just lost the genetic lottery with respect to the epigenetic clock. Actually, your epigenetic aging rate was faster, so paradoxical association. I want to conclude this presentation with 2 slides of conclusion. So conclusion slide number 1. Clearly, the epigenetic clock and methylation in general is an attractive molecular biomarker of aging. It's highly robust. I generated methylation data from blood tubes that were melted, okay? People wanted to throw them away, but you get very clean data. Yes, it's prognostic of mortality. It also allows you to contrast the ages of different tissues. For example, I mentioned female breast tissue is older than the rest of the body. I didn't mention the cerebellum tends to age more slowly. Most studies that used telomere length and other biomarkers can be revisited using these tools. There's freely available software on my webpage. It works on all Illumina methylation platforms. Conclusions number 2, big picture. These methylation clocks really reveal a deep connection between developmental processes, for example, fetal retina, and aging. And I mentioned the epigenetic clock theory of aging. I do want to emphasize there are several epigenetic clocks. I mentioned the pan tissue clock, the skin and blood clock, the Grimm age clock, and so on. So, and all, of course, correlate very strongly with chronologic age. However, they are more or less susceptible to endogenous and exogenous stress factors. Grimm age would relate very strongly to smoking. The pan tissue clock would not relate to smoking. We do have methylation age estimators that work beautifully in cell culture systems, fibroblasts, organoids, and so on. Having said all of that, I want to talk to people who design human clinical trials. Methylation age or any of these biomarkers will never replace standard clinical measures, blood pressure measures, lipid levels, cholesterol levels, glucose measures, CRP, and so on. They will add to these biomarkers, but they will certainly not replace them. When you do your own studies, you want to think very carefully about the source of DNA. Remember, when it came to female hormone therapy, we found very nice results in buccal swabs, but none in blood, And so think carefully about that. And I acknowledge the many people I mentioned during the talk, R.K. Liu, Morgan Levine, Austin Quack, and many other collaborators. Thank you so much.
You talked about you're getting lucky with your clock or unlucky, but you also talked at times about how lifestyle differences affect the clock and so on. It was unclear to me what are the relative magnitudes that come in here. What are the effects of lifestyle within a person on changing the clock? Could the clock, if there are lifestyle changes that can change the clock, that seems like that would be an early indicator and cheap indicator of predicting the outcome from some intervention. So, all the issues of causality are mixed in with this as we move across people or within people. Could you kind of comment on that general?
So epigenetic aging is a complex trait, very much like many other traits people measure. It very much is under genetic control. And to give you a number, roughly 40%, heritability is about 40%. Conversely then, 60% is not genetic, right? And when it comes to the question then of what's the effect of lifestyle, it very much depends on the organ we are talking about. For example, I mentioned that obesity is very much associated with epigenetic aging in the liver, strong effect. I want to say proportion of variance explained maybe 20%, it's a strong effect. However, if you ask whether obesity has an effect on aging in blood, barely noticeable, much weaker. Maybe it explains, 1% or 3% of the variance, So it's very much, you need to ask this question organ by organ, There are, when you are an aging researcher, you always like to have biomarkers that are very susceptible to good lifestyle choices. Why? You may have an intervention program and you want to convince people to lose weight, or you want to convince them to eat vegetables. And you would like to say, look, 3 months after you ate vegetables, look, you're now a little bit younger, And in certain ways, certainly my pan tissue clock does not do that for you. Why? Because it really relates to these innate aging processes that you cannot cheat by lifestyle interventions. If you want to, Grimm age is much more susceptible. So I think there's evidence, even unpublished though, but anecdotal evidence that weight loss is associated with slower Grimm age. So stay tuned for publications. But in general, if you want to show that the lifestyle choices have a good effect, go with these standard measures. Just measure glucose levels, right? Show people who don't eat junk food, their glucose levels improve, So that's what I would do. My personal goal was always to invent biomarkers and tools that allow you to study these innate aging processes that really relate to that are not less susceptible to lifestyle choices. Yeah, other questions?
Well, okay, it was kind of goes with that, but you showed some epigenetic clocks that were developed in animals, and are you aware of any trials that have been done with, in addition to intervention, like rapamycin or metformin, or even like social isolation or things like this?
Yeah. I certainly am currently involved in human clinical trials where we evaluated various interventions, and we actually have some very nice results for a particular intervention I cannot talk about right now. But there are interventions in humans that affect epigenetic age. Stay tuned, Will come out in a month or so. When it comes to rapamycin, as you may know from others, there are people who study rapamycin, certainly in dogs. That's an exciting study. And every time I meet these people at conferences, I beg them to evaluate the blood methylation levels. I've certainly just a week ago sent off another email saying we have a methylation clock in dogs that works very well. And anyway, so I'm very collaborative. Anybody who has an exciting clinical trial or an exciting animal model, approach me. I will help you. But yeah. So we have epigenetic clocks in any animal you can think of: rat, vervet monkey, baboon, chimpanzee, marine mammals, beluga whales, dolphins. I don't know any opossums. So horses, sheep. So yeah, whatever you can, as long as it's a mammal, I can help you. Yeah. Other questions? Great. Thank you so much for your attention. Yeah. Bye.